particularly the sodium ions, will reach an equilibrium state at a certain potential level. Beyond that, it will not produce higher threshold action potential. So that's the reason, even if you apply a maximum stimuli directly, either directly to the muscle or directly to the nerve fibers, okay, it will produce an action potential which is similar to the threshold stimuli. Okay, so then depends the duration. All the tissues respond to a specific intensity for specific duration. It varies in different tissues. Okay, so based on that, the strength duration curve we can draw. So Rio based chronaxy. Later we will discuss about this. Okay, so this is important to understand. Channels. So what is the channels here? There are different types of channels are present in the nerve fiber or in the muscle membrane. There are different types of nerve channels present. What is channel here? They are nothing but the proteins synthesized in the from the nerve tissue or from the muscles inserted into the membrane. Okay, so there are leaky channels. Say for example, if you take the cation leaky channels, it will allows all positively charged ions to enter, to move in or move out of the muscles, muscle tissue or nerve tissue. So all the time they are open. Now, when all the time they are open, will they allow all the ions to move in, move out? It depends what type of channel it is. If it is cation channel, it will allow only the cations. If it is anion channel, it will allow only the anions. So there are presence of leaky channels, and more probably, and most of the time, leaky channels allows potassium to move out of the cells. So whenever a channels get open, you must understand, you must know about which ion will move, whether inside or outside. Why I'm saying leaky channels are mostly permeable to potassium ions, because potassium, they are more permeable than the sodium or chloride or any other ions. Okay, mainly if you look at the in the involvement of movement of ions during this resting membrane potential or action potential, mainly sodium and potassium it involves. Okay, so potassiums are more permeable than the sodium ions. Okay, then the another question arises: why potassium is more permeable than sodium? So it depends the hydrated form. The hydrated form particularly depends the mass of those ions. Even if you look at the sodium and potassium, sodium mass of the sodium is more than the potassium. But in the hydrated form, usually when in the liquid or in a fluid, they will remain in the hydrated form. In hydrated form, mass of the sodium is more than the potassium. Molecular weight of the sodium is more than the potassium. So potassiums are easily allowed than the sodium ions. So there are presence of gated channels, what we call it as voltage gated channels. So I would have read about all this. Okay, I'll just skip about this. Then ligand gated channels are chemical where the neurotransmitters act on this. Okay, then yeah, this terminology you know about the polarization, depolarization, repolarization, hyperpolarization. I'll just skip this and then I will continue. What is that graded potential or local potential? So a local changes in the membrane potential that occur in varying grades or degrees of magnitude of strength. Okay, varying grades or degrees we can say, or strength we can say. Okay, so usually if you look at the graded potential from the resting membrane potential, it can go up to -10. A 10 millivolt change you can find out, or 20 millivolt change you can find out. Okay, so beyond that, if it goes beyond that, then it will reach to the threshold level, then it will produce an action potential. Okay, so it can be like the graded potential can be in the depolarizing direction, or it can be in the hyperpolarizing direction. It depends what type of channels it opens. Okay, in the synaptic membrane, in the synaptic junction, depending upon the neurotransmitter get released, you can find out excitability or inhibition, right? So if there is a excitation, then there must be opening of sodium channel. If there is inhibition, then there must be opening of potassium channels. So opening of sodium channel causes depolarization. Opening of potassium or chloride channel causes hyperpolarization. Depolarization important to produce an action potential. Initially, in the depolarizing direction, there will be opening of the sodium channel. So it produces a depolarization. If it reaches to the threshold level, it will produce an action potential. I will show it. I will show this in the diagram. Okay. If there is opening of potassium or chloride ions, then that will cause hyperpolarization. There will be a downward deflection. You can find out. So graded potential will travel to a shorter distance and then it will die out. So this is what happens during this graded potential. The neurotransmitter, the acetylcholine, acts on the acetylcholine receptors. Okay, so when it acts on the acetylcholine receptors, there will be opening of sodium channels. Okay, so the sodium ion will enter into this. So when the sodium ion enter, since the positive charged ions are entering, inside will become positive. So this is what we call as a depolarized. Okay, so that particular area is depolarized. So a local change in the polarization is produced. That's why it is called as local potential. So since whether we can is it possible to obtain a different grade, means a 10 millivolt change, a 15 millivolt change, an 18 millivolt change. Yeah, that is possible to obtain depending upon the neurotransmitter get released and number of ligand gated channels get opened. So that's why it is called as graded potential. We can grade it. With that local potential, we can able to grade it. 5 millivolt change, we can bring it. 10 millivolt change, 15 millivolt change. Okay, so that's why it is called as graded or local potential. So it travels on either side direction, but it will die out if there is no further stimulus. Okay, so this is what in this diagram you can see. When you apply the stimulus, a depression is produced. The same stimulus, not the same stimulus, but if there is a release of different neurotransmitters, it can produce hyperpolarization. So depolarization and hyperpolarization depends upon the type of channel open. Whether it is sodium channel is open, you can find out depolarization. If the chloride or potassium channel is open, you can find out hyperpolarization. So depolarization produce later it can produce an action potential. Hyperpolarization, it will produce inhibition. Okay. Now, if the stimulus is weak, a small changes you can find out in the local potential. If the stimulus is strong, you can find out here further changes. So small changes, weak stimulus, a small changes, maybe 5 millivolt change, or it can bring 10 millivolt change. Okay. So if you measure it, the graded potential, you can find out that slowly it will die out. So you can see depolarization. Initially, it can bring 10 millivolt change. As it travels, it will become 5 millivolt change, and then it will die out. Okay. So this potential, this local or graded potential, can be summated by repetitive stimulus. Okay. Suppose if you are applying a subthreshold or subliminal stimulus, when by applying that subliminal or subthreshold stimulus, it can be summated. If you apply continuously, repeatedly, okay, temporal summation happens. Okay, all this what I am talking is related to electrical potential changes, not the mechanical event. Electrical event I am talking. Okay. So that electrical event can be in the muscle tissue, cardiac muscle tissue, or skeletal muscle tissue, wherever it may be. Okay. So repeated stimulus will produce a temporal summation. Stimulus simultaneously by different modality, or in case in body by using different nerve terminals. If there is a simultaneous stimulus, then that produce spatial summation. Okay, so that is the difference between temporal summation and spatial summation. Now, all this summation or the stimulus which produce the local potential. If it reaches to the threshold level, now what is the threshold level? A 15 millivolt change or a 20 millivolt change from minus 70 in the nerve tissue considered as resting membrane potential. From the resting membrane potential, if there is a change for 15 millivolt, if you bring it minus 55 millivolt, then it can produce an action potential. So that change my 15 millivolt change or 20 millivolt change will vary in different tissues. So graded potential, you can find it at the receptor levels. Here we call it as a receptor potential, and the synapses, here we call it a synaptic potential. So depending upon the neurotransmitter get released, we can find out inhibitory postsynaptic potential. Say for example, GABA will produce inhibitory potential, or if there's a glycine, or if there is a release of acetylcholine, it will produce excitatory postsynaptic potential. So in the neuromuscular junction, this local potential, we are what we call it as EPP, end plate potential. Okay. Now for action potential, this is what exactly the action potential. So when you go through the books, you can find out this kind of recording. You can find out this is the experimental recording. Okay. So experimentally, what they will do, the nerve fiber, you can see a stimulator here. They will place the stimulator, and then they will stimulate by using the stimulator, and then you can find out the production of action potential recorded by using cathode ray oscilloscope. Okay, so in order to record action potential, you can see the placement of these electrodes. One electrode is above and another electrode is inside the nerve fibers. Then only we can record the action potential. Whatever we obtain by using the Serrington drum or Serrington experiments, all these are mechanical events. Okay, so in order to obtain the electrical event, you have to insert the electrode into the muscle tissue or into the nerve fibers. One electrode outside, one electrode inside to obtain the potential difference. Okay, suppose in case if you place both the electrodes outside, is it possible to obtain the action potential? Yes, it can. It is possible to obtain the action potential, but the difference is it will be biphasic. Now, mostly the action potential recording is monophasic. If you place the electrode, both the electrodes outside, then it will become biphasic recording. During depolarization, there will be upward deflection. During repolarization, there will be downward deflection. But in monophasic, during depolarization, you can see upward deflection. Then during repolarization, it will come back to resting state. Whereas in biphasic, what I am showing here is monophasic action potential. In biphasic, the repolarization will go downwards like this. Okay, so like how we are looking the spike upward direction. Similarly, the spike you can see in the downward direction. That is what we call as biphasic action potential. So, is it possible to record biphasic? Of course, it is very easy to record the biphasic. Usually, we do in clinical site. What is that? It is nothing but ECG. Okay, electrocardiograph rhythm is a biphasic action potential. What we are recording there. Okay, monophasic is it's little difficult. It requires invasion of electrode. Okay, so it is difficult. In animal experiments, it is possible to do it. So in an action potential, you can find out from the starting. It is obtained from the nerve fibers. So minus 70 is the resting membrane potential. Okay. Stimulus artifact. What is that stimulus artifact? Is nothing but as soon as you place the electrode, you can find out a small deflection there. This is what we call the stimulus artifact. Okay. Then if you apply the stimulus, the application of stimulus and then production of action, production of a change, the production of local potential. So what we call it is latent period. Okay, so then you can find out action potential there. Okay, full-fledged action potential over here. All right. So we will discuss each and every phase of this action potential. Okay. All right. Now here, understanding of the ionic basis. Why I am taking ionic basis today? What's the reason for this? if you understand this ionic basis, we can understand what is absolute refractory period, what is relative refractory period. So can I stimulus fiber during the relative refractory period? So it is possible to stimulate during relative refractory period, but not during the absolute refractory period. So I hope that understanding will become quite easier when you go through this ionic basis or molecular basis of this action potential. Okay. So at the resting state, this diagram shows during the resting state. Okay. First, I will explain what is this gate over here. So gate here nothing but the sodium channel, and here you can see the potassium channel in the cell membrane. Okay. So the sodium channel, if you look at, there are two gate system. Okay, activation gate and inactivation gate. Whereas for the potassium, you can find out only one gate. All right. So that's the difference between the sodium and potassium. Okay. What is this gate system? It is nothing but when there is change in the membrane potential, the protein, a change in the configuration of protein happens there. Okay. So, right now, remember, there is activation and inactivation gate for the sodium channel, and there is only one gate for potassium. Now, sodium ion is responsible for this depolarization of the action potential, whereas potassium is responsible for the repolarization of the action potential. So, at the resting state, they are in the normal position. So, all voltage gated potassium and sodium channels remain closed. So, when the activation gate is closed, now we can see the activation gate, it is the closed position. Even for the potassium, it is in the closed position. This is at the resting state. Now, so at the resting state, the sodium or potassium cannot enter into it. Or as I told earlier, whenever you say at the opening of ion channel, whether there will be entry of an ion or it will go out of the cell. That you should understand. So to understand that, you must know about the concentration of the ions inside and outside the cells. So sodium ion concentration outside the cell is 145 milliequivalent per liter, whereas inside it is 15 milliequivalent per liter. So sodium ion concentration outside is more than the inside. Okay. Whereas for the potassium ion, the potassium concentration inside is 150 milliequivalent per liter. Outside is 5 milliequivalent per liter. So inside concentration in potassium is more. So whenever there is opening of the channel, so automatically from higher concentration to lower concentration, ion will move. So for always, whenever there is opening of the sodium channel, there will be movement of ions, movement of the sodiums from outside to inside, influx will be there. Whereas for the potassium, whenever there is opening of the potassium channels, okay, there will be movement of potassium from inside to outside, efflux will be there always. So it is based on the concentration gradient, not only the concentration gradient, electrical gradient also inside the cell. Okay, the inactivation gate is inside the membrane, whereas the activation gate is outside the membrane. Normally, inside it is negative, outside it is positive at resting state. Now, to understand this resting membrane potential, you have to go through the ionic basis for this, mainly potassium and sodium potassium channel activation pump, what we call the sodium potassium pump or sodium potassium ATPase, plays an important role for this resting membrane potential. So for resting at resting state, now fibers, if you take, it is minus 70 millivolt in terms of the voltage inside the cell, it is minus 70. So we say resting membrane potential is minus 70. So outside you can see plus, okay, inside you can see minus. Okay. So this electrochemical gradient, based on that electrochemical gradient, there will be movement of ions. Okay, now resting state, the sodium ion is concentrated outside more, so it will remain outside, and the potassium inside, the potassium ion concentration inside it is more, so it will remain inside. Now, when you apply the stimulus, okay, what happens there? So when the adequate stimulus is applied, that is the threshold stimulus is applied. So when I say threshold, at least it should bring a change, okay, 15 or minus 15 or 20 millivolt change it should bring from minus 70. So this the -50 here you can see the dashed line you can see in the diagram what is called as a threshold level or firing level. Threshold level or firing level. So when the membrane potential reaches at this level, at this firing level, okay. So till that what happens, there will be a change in a small change, the local potential happens. When that local potential reaches to the -50 millivolt, there will be opening of this sodium channel. What is that opening of the sodium channel? Now you can see the activation gate is opened in the diagram up in the up diagram. In the diagram above, you can see the opening of activation gate. So if there is opening of activation gate, what happens? So there will be entry of sodium ions. Okay, so when you apply the threshold stimulus, adequate stimulus, so there will be opening of this activation gate, sodium ion enters. So when there is entry of sodium ions, what happens? You can see the membrane potential reaches to the 0 millivolt. Membrane potential now it has become 0 millivolt there. Okay, now in this above diagram you can see plus minus equally, right? So since the positive charged ions are entering inside, it has become positive inside. Okay, 0 millivolt it means there is no voltage difference between the across the membrane. Okay, so these ions further kept open for some time. Till how long it is kept open? The sodium channels are kept open up to plus 30 millivolt. At 30 millivolt, the sodium ion cannot enter inside. Why? The sodium ion, the inactivation gate, you can see the another gate closed. Because of this closure of inactivation gate, the sodium ion cannot enter inside the cell. At what level it happens? That is the peak of the action potential, what we call it as. Okay, so the potential is reversed means what? Now at the peak of the action potential, inside it has become plus 30 millivolt. So whenever we talk about the change in the membrane potential in terms of inside the potential, what happens? Based on that, we say there is a negative, there is a positive, not outside. Please remember that. So the potential earlier inside it was negative, now outside it has become positive. Positive, the plus 30 millivolt. At this stage, the inactivation gate closed, sodium ion cannot enter. Okay, why there is closure of this gate system? Because at this level there will be a reaching of saturation of what we can say equilibrium level of sodium entry. Okay, so mainly this voltage gate, that's why we call it as voltage gate, responds to a different potential level. Okay, at threshold, at firing level, from the threshold level to firing level. Okay, that is from the resting membrane potential level to firing level, from -70 to -50. When the potential reaches to -50, that is the firing level, there will be opening of the activation gate, sodium will enter. When the potential reaches to plus 30 millivolt, there will be closure of the inactivation gate, sodium cannot enter. So the opening of this or closing of these gates depends upon the change in the membrane potential. So there will be change in the protein configuration, thereby there will be opening or closure of these gates. Okay, so at this potential at plus 30 millivolt, there will be opening of potassium gate. Now potassium can enter. Okay, so now you can see the potassium moves out, what we call it as potassium efflux. So since a positively charged ions are moving out, so what happens here? The negativity inside will become less and less and less. So if you obtain the recording, you can see here again the membrane potential will return. Means inside slowly will become more negative because positively charged ions are moving out. Okay. Now there will be continuous outflow of efflux of potassium ions. Okay. the efflux of potassium, if you look at what we call this potassium conductance, okay, efflux of potassium takes a longer duration than the influx of sodium. Okay, so the the it the membrane potential, even though it reaches to threshold level, okay, we can see there will be continuous movement of this. When the membrane potential reaches to minus 50 millivolt, please remember the sodium gate, if you look at, uh, will come back to the original resting state. When the membrane potential reaches to minus 50 volt, from plus 30 to minus 50. That is what we call as repolarization. Now polarization is become reverse, repolarized. From the chains, it come back to normal state, repolarized. Okay, so when it get repolarized, the at minus 50 millivolt, the gate will come back to normal state. Whereas potassium continues to efflux out. Okay, when the potassium continues to efflux out, so that will produce a hyperpolarization. The membrane will become hyperpolarized. Why we say hyperpolarized? Because continuous efflux of this potassium out inside will become more negative. So the recording, if you look at it has gone below minus 70. Minus 70 is the resting state. It has gone below the minus 70. So at this state, we call it as hyperpolarized. Okay. So that is the reason for the hyperpolarization is continuous opening of the potassium gate. More potassium efflux will be there. Okay. So at hyperpolarized state, once it reaches to the hyperpolarized state, the potassium channel will come back to the resting state. Once it come back to the resting state, then the membrane potential slowly will reach to resting state. Okay. So at this state, now whatever the movement of ions, some amount of sodium is entered, some amount of potassium has gone out. Now that ionic concentration has to be restored back. Okay, the sodium which has been entered inside will not remain inside. Okay, after some amount of stimulus. Okay, not for a single stimulus, but after some amount of stimulus, the sodium which has moved out, the potassium which has moved sodium which has moved in and potassium which has moved out will come back to the resting state by sodium potassium pump, ATPase. Even why that pump is required here? Okay, even after depolarization, entry of sodium inside and the movement of potassium out, still the concentration of the sodium inside is less and the potassium outside is um, the normal concentration of sodium, if you look at inside is less, outside is more. Normal concentration of potassium inside is more, outside is less. Even after the movement, that concentration remains same. That's why we require a sodium potassium pump here. Moves the 3 sodium ion outside the cell membrane, okay, and moves 2 potassium ion inside the cell membrane. You can see the difference here. 3 positive charged ions moved outside, 2 positive charged ions, that is potassium, moved inside. This itself will bring a negative potential inside. Okay, so the sodium potassium pump will try to restore the sodium ion entered in and the potassium ion has gone out. Okay, so this is what happens at resting state. So this is the ionic basis of this action potential. Okay, now based on this, if you look at this diagram, you can see the action potential about then you can see the sodium conductance, movement of sodium ions, and then the potassium conductance. Okay, potassium conductance is slow and longer duration. That's why there's hyperpolarization. Okay, so this is the recording of action potential. This is the ionic basis of action potential based on the type of nerve fibers, whether if it is whether you are recording monophasic or biphasic, or if it is a combined neurons, whether sensory and motor neurons, or within the neuron itself, there will be different thresholds. Threshold will be there, so you can get a compound action potential. Okay, so when you go through the books, different type of terminology they will use. So the spike you can find out. Okay, uphill movement, downhill movement. Sometimes you can see overshoot above from 0 to 30 plus 30 millivolt. They call it as overshoot. Okay, so often after depolarization, after hyperpolarization, they use different terminology. But if you understand the ionic basis of it, it's fine. Okay, this is how exactly recorded. This is the biphasic. ECG is the one of the biphasic recording. During depolarization, there will be upward deflection. During repolarization, there will be downward deflection. All right. So this is the compound action potential. A mixed nerve fibers, we will get the compound action potential. Okay. Now we will come back to this later. What's the refractory period? Absolute refractory period and relative refractory period. There are two types. So during the action potential, a second stimulus, no matter how strong, will not produce a second action potential. A second stimulus, no matter, even if you apply the suprathreshold stimulus, you will not get an action potential. This means the nerve fiber or muscle tissue is in absolute refractive state. Refractive means a period of inactiveness. There won't be any response. So the voltage gated sodium channels enter closed inactive state at the peak of the action potential. So in order to produce an action potential, what you observed now, there will be depolarization, opening of the sodium channel, and there will be depolarization, right? So the sodium channels goes into a inactive or closed state at plus 30 millivolt. Okay. So during repulsation, the sodium channels will come back to the resting state. Okay. When the sodium channels are closed state, if you apply a second stimulus during that state, you cannot bring the action potential there, because the sodium channels are already in the inactive state. It is closed. You cannot bring a response there. So this is what we call as absolute refractory period. Now, in this diagram, you can see the absolute refractory period. The red color, you can see the
Ramesh, Babu.
Yeah.
Can I just interrupt the previous slide?
Yes.
Yeah. So here, when you mean a closed inactive state, it means the gate inside is closed, right?
Yes, of course.
Okay, right. That's what I wanted.
That inactive gate will be closed.
Closed. Okay, fine.
Yes.
Thank you. Yeah, please continue.
Yes. So this membrane potential, now you can see absolute refractive period, time duration. If you look at in the action potential curve, the first, the red color line you can see there, that is the action potential. Green is the sodium conductance, and blue is the potassium conductance. Okay, the red color line which indicates the action potential. If you look at from the starting of the stimulus to the 1/3 or half of the refractory period, entire depolarization phase, and half of the repolarization phase. Okay, up till that you can see the absolute. If you apply the second stimulus during this, whether it is a suprathreshold or supramaximal stimulus, there you will not obtain the response. Now, what is the relative refractory period? Now, during this relative refractory period, the membrane, the sodium channel will come back to this resting state. Okay, some of the channels, the sodium channels will come back to the resting state. Okay, so during this state, if you apply the second stimulus, now say for example, the imaginary line for the absolute refractory period where it ends at that line. Okay, and below that you can see from there to the refractory period. Now, during this time, if you apply the stronger stimulus or suprathreshold stimuli, then you can obtain a second action potential. Why we required a suprathreshold stimulus? Because already the membrane is in a repolarization state. Potassium ions are moving out. Okay, the sodium ions are, so the sodium channels are returning into the rest, the previous state. Okay, now it is possible to open the sodium channels, but it has to come across this repolarization, because already the potassium channels are open, repolarization is happening. Okay, at this time, if you want to open this sodium channel, and extra strong stimulus is required, supramaximal suprathreshold stimulus is required to open that sodium channels. Then you can obtain one more action potential. So that's why it is called as a relative refractory period, not the absolute refractory period. Okay. During this relative refractory period, if you apply the suprathreshold stimuli, okay, we can apply an action potential. One more response, you will get it. Okay. Whatever I am talking now, please remember it is electrical event, not the mechanical. Later I will come back to the mechanical event. Okay. So this is now in this diagram you can see the correlating electrical event with the mechanical event. Now, if you look at the application of the stimulus, okay, normally whether you stimulus the muscle directly or nerve fibers, you will obtain this response, but the time latent period will differ. Okay, if you apply stimulus to the nerve fiber, there will be release of neurotransmitters, then there will be opening of ligand-gated channels, and then that membrane potential has to reach the this threshold level, and then there will be production of action potential. The latent period duration is slightly longer. If you apply the stimulus, electrical stimulus directly over the muscle, okay, normally in our body it will not happen. But if you apply the stimulus experimentally directly over the muscle, the latent period will be shorter. Okay. Now, in this diagram, if you look at down the diagram below, you can find out the mechanical event because of this action potential, because of this electrical event. So there is action potential, and then we can find out the mechanical event. Now, if you draw the diagram, imagine a line from the peak of this action potential down the muscle, where the mechanical event you get. Okay. Now you can see the difference during the depolarization phase. Okay. During soon after this depolarization phase, okay, you can find out starting of the contraction. Now, if you draw the imaginary line from the 1/3 or half of this repolarization phase to the muscle response, you can see only a smaller portion of the contraction phase will come under this relative refractory period. Okay, most often after depolarization, you can find out starting of the contraction. Okay, then the contraction phase. So during this contraction, the muscle will undergo repolarization. Okay, so that we can see half of the contraction phase, 1/3 of the contraction phase from the beginning, 1/3 of the contraction phase will come under the relative refractory period. So during this phase, if you apply the second stimulus with the higher strength, you will obtain an additional action potential and additional muscle contraction. So if you apply the electrical stimulus during this during this relative refractory period with higher stimulus, you can obtain a response. Okay, so if you apply the second stimulus during this absolute refractory period, you will not obtain any response. So that's the difference. Okay, so this later I will explain how the tetanic type of contraction is obtained. Okay, so the time is now almost going to 10. I will finish it now. So this is the relative. It tells the same thing about the relative refractory period. Okay. So what is the advantage of this? The refractory period limits the number of action potentials. They also increase the reliability of neural signaling by limiting the extra impulses. So these are the advantage of this relative refractory period. So now fiber or muscle tissue should undergo a relative refractory period so that smooth coordinated muscle activity we can obtain it. Okay. Whereas in the same thing, the cardiac muscle, if you look at, just for comparison, I have given here, the action potential in the cardiac muscle has a longer duration, nearly 300 milliseconds. Whereas in the skeletal muscle, it is 2 milliseconds. Here it is nearly 300 milliseconds or 250 milliseconds. Okay, absolute refractory period, if you look at, it is longer than the relative refractory period. And the red line, if you look at, the red line indicates the contraction and relaxation of the cardiac muscles. Most of the contraction phase will come under the absolute refractory period. A small part under the relative refractory period. So during relaxation of the relaxation of the cardiac muscle, the relative refractory period lies, right? So if you apply suprathreshold stimulus, you can obtain extra systole there. But if you apply during the absolute refractory period, where most of the contraction phase has been occurring, you cannot obtain the any extra systole, extra contraction. So what's the benefit of it? So the cardiac muscle, in order to tetanize a muscle, in order to get tetanic type of contraction, you have to apply the stimulus during the contraction phase, not during the relaxation phase. In order to get the tetanic type of contraction, you have to apply the stimulus during the contraction phase. Now, the skeletal muscle, you can apply the electrical stimulation during the contraction phase because it will undergo refractory, only relative refractory period, and then normally you can see the contraction and relaxation response, right? So, if you apply the stimulus during the contraction phase, it is possible to apply a stimulus during the contraction phase, and you can find out the tetanic type of contractions. Whereas in the cardiac muscle, you cannot find out the tetanic type of contraction because even if you apply the stimulus during the contraction phase of the cardiac muscle, it will not respond. So that is one of the advantage of this refractory period. Okay, but the duration of this refractory period in the cardiac muscle, in the skeletal muscle differs because you can see the action potential of the cardiac muscle has a longer duration than the action potential of the nerve tissue or skeletal muscles. Okay, so this is the important of this about this. So if I get time, next time I will correlate with that serotonin reflex. Okay. Are you able to understand? You can, if you have any doubt, you can ask me now. Because time is almost over.
That was a very, very clear explanation of many things. So I'm really very, very impressed with the minute details which you have put in, which throws a lot of light. I'd like the participants to just give their opinion about both the presentation as well as any clarifications you can please ask.
Sir, Ramesh sir.
Yeah, please, yeah.
So very, very excellent presentation, sir. I have never seen like this. Nobody taught us physiology like this in our college.
Okay.
I would appreciate for this clear and minute details, which we really understood the movement of ions today.
Okay,
it's very informative.
Yeah.
Sir, I have a doubt.
Yes, sir.
You said there is a outside there is a positive voltage and inside
voltage
it
changes.
is negative.
Inside it is negative,
negative,
outside positive. Yes.
So and we have a sodium outside and potassium inside, correct?
Yes, sir. Yes, sir.
Both are positively charged. You know how
Positively
there is a
charged.
difference.
Yes,
how
sir.
there is a
Yes,
difference.
Yes,
Negative
sir.
and
That
positive.
part, yeah. For that, we must understand the ionic basis for the resting membrane potential, sir.
Yeah.
How the resting membrane potential is generated for the given concentration of the ion. So most probably same like that action potential, how it is generated, what I explained. Similarly, it depends more on potassium ions.
Okay.
So that 150 concentration inside and the 5 concentration outside, it all depends after applying minus 70 millivolt. Okay,
Okay.
it generates that concentration difference. Actually, suppose if you take only the potassium, how long a potassium can move inside and outside the cell? Um, and if you take the sodium, how long the sodium and potassium can move inside and outside the cell? Both will reach an equilibrium. At that equilibrium state, the voltage difference across the cell membrane is minus 70 millivolt. So
Okay.
since now already time is 10, next time I will explain about that.
Sure. Sure. Thank you,
Yes.
sir. Thank you.
Yeah. Thank you.
Others, you can just tell your opinion.
Electrodes are placed superficial. Yes. Yeah. Depends why biphasic when both electrodes are placed superficial and monophasic when one electrodes. Yeah. Uh, so biphasic action potential. If you look at when we are placing the electrode, both the electrodes outside the cell membrane or nerve tissue. Okay, you are stimulating the nerve tissue. Now there will be change in the membrane potential. So there will be depolarization and then repolarization. Now there are 2 electrodes are here. The depolarization wave will cross the 2 electrodes. First electrode it will cross, then it will reach the second electrode. Okay. Now when it cross the first electrode, the change in the potential difference between the first and second electrode will be recorded. That will be recorded as a upward deflection during depolarization. When the muscle completely become depolarized, now there is no difference between first and second electrode. There is no voltage difference between first and second electrodes. So the muscle will not, you cannot find out any recordings. So the upward deflection will come back to resting state. Now, when the repolarization starts, now you can find out the difference between the first and second electrode during repolarization. So you will see a downward deflection. Now, if it is completely repolarized, you won't find out any difference between the 2 electrodes. There is no potential difference between 2 electrodes. So it will come back to the resting state. This is how we obtain the biphasic action potential based on the difference between 2 electrodes. Whereas monophasic, one electrode is outside, another one is inside. At the beginning itself, it will show the difference. What's the difference? That is the resting membrane potential. So you will obtain the resting membrane potential through this monophasic. Now, when you apply the stimulus, when there is a change, you will record upward and then downward deflection as a repolarization. So that's the difference. Any other doubt?
So I'd
Yeah,
like
please.
Sajju, Riddhi to give your opinions.
Yeah.
Hello.
Please tell me.
Yes, so it was a very detailed explanation.
Thank you.
That those action potential and the phases of action potential and how the ions will be transmitted, everything was very clear.
Yes.
So, thank you for the explanation.
Oh, yeah. Thank you very much.
Okay, Ramesh, I think it was,
Yes, please.
it
Sure.
was
Yeah,
very,
already it
very
is 10, I
detailed.
think. Sorry for a long time.
No, that's okay, right? I think it will save a lot of the time at a later point of time. So that's how I see it. Okay. Right. So
Yeah,
thank
sure.
you so much. And I think we will have the continuation of it in the next part. And
Yeah, sure, definitely.
and after this presentation, I'm also able to correlate and see in the during the reflex conduction how it is
Definitely.
different.
Next time I will do that.
That part I'm able to better understand that, because
Yes.
when Sherrington says only about the reflex part, right?
Yes. Yeah.
Understanding this becomes very important. So thank
Yeah.
you so much, Ramesh.
Yeah.
Once again, thanks from our side, from every one of my students. Thank you.
Thank you very much. Thank you.
Um,
Thank you for giving
thank
this opportunity,
you.
Roshan.
Yeah, that's my pleasure. And we'll meet coming Tuesday. But I think before that, we'll also be meeting on Sunday for the other one, which is going to be on the some of the physiological basis in development of locomotion, what differences are happening. Right? So that
Okay.
we'll be looking into
Sure.
it. Okay?
Yeah.
Yeah. Thank you so much. We'll end the meeting here.
Okay. Yeah. Thank you.
Thank you, sir.
Thank you, sir.